Okay. Welcome to Zoology Classroom. In today's class, we are going to learn about open and closed circulatory systems and myogenic and neurogenic hearts. First of all, let us know what is the importance of circulatory system, why we need a circulatory system. Circulatory system has developed in higher level animals. We know that every cell requires food and oxygen, and they will utilize these food particles and oxygen in various metabolic activities. As a result of these various metabolic activities, what happens? As a result of these various metabolic activities, several waste materials will be generated in the cells. And all those waste materials have to be eliminated from our body. So here different functions are there. So as animals have grown in size, different functions, different types of organ systems have been developed in higher animals. So for example, to supply nutrients and food, digestive system is there. To supply oxygen, respiratory system is there. So this blood or circulatory system only collects nutrients or fuel molecules from the digestive system, oxygen from the respiratory system, and supplies all these materials to remaining all body parts. Likewise, as a result of these metabolic activities, all the cells, they will generate carbon dioxide and nitrogenous waste material like ammonia, urea, or uric acid, etc. And all these things have to be collected from those organisms, and those materials have to be sent out of the body. So here in this diagram, we can see that the blue thing, this blue pipeline system, this and some part it is having some reddish color. So this is the circulatory system. So what circulatory system is doing? Circulatory system is collecting nutrients from this digestive system, and it is supplying all these nutrients to the cells. Likewise, it is also collecting oxygen from these lungs, and it is transporting or supplying all those things, all those oxygen molecules to the cells. Likewise, in return, what it is doing, it is collecting nitrogenous waste materials and carbon dioxide. And carbon dioxide, after collecting from the cells, this carbon dioxide will be given to the lungs to send out of the body, and nitrogenous waste materials will be given to this excretory system to send out of the body in the form of urine. So this is the function of circulatory system. If circulatory system is not there, we cannot receive the digested food materials. We cannot receive oxygen. All cells, they cannot receive all these molecules, and cells cannot get rid of the waste materials like carbon dioxide and nitrogenous waste materials also. So this is the importance of circulatory system. That's why we need circulatory system to be functioning normally at every time. If the circulatory system stops to function for 5 minutes of time, we cannot survive. So that is the importance of the circulatory system. So then in the animal kingdom, two types of circulatory systems have been developed. First one is open circulatory system. This open circulatory system can be seen in arthropods, some molluscans, and some echinoderms. So what is the difference between open circulatory system and next one is a closed circulatory system? Open circulatory system, another one is closed circulatory system. So I have forgot to place the another slide, closed circulatory system. I will explain it later. Okay. So coming to open circulatory system, in open circulatory system, you carefully observe this diagram. So in this, this is the heart, and this heart is collecting blood from some collecting ducts, and it is supplying blood to some other pumping ducts. So here, in our case, in open circulatory system, we will be having hearts, arteries, and veins. So veins will be collecting blood from the various organs. Arteries will be supplying blood to all other body parts. And these arteries and veins are connected with each other. They are connected to each other with blood capillaries. So nowhere they are separated. So, but here in open circulatory system, the blood vessel that is pumping blood to various organs is ending abruptly or bluntly. So you here in this diagram, you can see that this blood vessel coming from heart, it is bringing blood from the heart and it is opening into the open space. This is the open space. So the blood vessel is opening into the open space. So blood is coming out of the blood vessel. Okay, so and the blood is coming into the open spaces. And later again this blood will be collected. Again this blood will be collected by different other blood vessels, and these blood vessels, they will send the blood to this heart again. So in this way, here in this diagram, we can clearly see this. This is the first one is the open circulatory system. Second diagram is of closed circulatory system. So this is the heart. So heart is supplying blood to the tissues through arteries. Okay, these arteries are bringing blood to the tissues. After coming to the tissues, the artery is opening through holes. So the blood is coming into the open space, which is surrounded the tissues, organs. So blood is coming out of the circulatory system. If we dissect the body of a open circulatory system animal, we can clearly see the blood. Okay, but after exchange of materials taking place, once the exchange of materials is taken place, then what happens? The blood again enters into these blood vessels. These are venules. So, and again, these venules they are open through holes. They are opening bluntly. So these open venules, they will collect blood from these tissues and they will send this blood to this heart. So in this way, in open circulatory system also, heart is there, pumping vessels are there, collecting vessels are there. Pumping blood vessels or supplying blood vessels and collecting blood vessels are there. But these supplying blood vessels and collecting blood vessels are not connected with each other. They are separated. As they are separated, blood is coming out of them and it is filling the open spaces. So in cockroach circulatory system also, we have seen that several hemocoelomic cavities are there. In the head region, cephalic cavity, and in the abdomen region, visceral cavity, and around the nervous system, perineural cavity. So like this, several hemocoelomic cavities are there. So the blood enters into these cavities. From these cavities, the exchange of materials will be taken place in between the blood and uh, tissues. So here we should not collect the blood of these animals as blood. Why? Because we know that in organisms which are having which are having closed circulatory system, in them blood will be always circulating inside the blood circulatory system only. Blood will never come out of the this system, circulatory system. And outside the tissues, we have tissue fluid or extracellular fluid. Okay, tissue fluid is there or extracellular fluid is there. If the tissue fluid is excess in quantity, the excess tissue fluid will be sent into another system called lymphatic system. Another system called lymphatic system. So in lymphatic system, lymph fluid will be flowing or lymph fluid will be circulating in lymphatic system. In practice or in real, there is no difference between lymph liquid and tissue fluid. Tissue fluid and lymph fluid are no different. The only difference is the placement. If the fluid is outside the tissues, that is called tissue fluid. If the fluid is present in the lymphatic system, then it is called lymph fluid. Okay, so that is the difference. Depending on the place where they are, the liquid will be called tissue fluid if it is present. If it is present inside the outside the cells or in the tissues, it is called lymph fluid if it is present in the lymphatic system. So here, if the lymph fluid, if the tissue fluid is excess in quantity, the all excess tissue fluid will be sent into the lymphatic system. In any case, tissue fluid is low in quantity, then what happens? lymph fluid from the lymphatic system come out and fills the tissues, and it makes up the quantity of the tissue fluid. So whenever there is excess tissue fluid, that will be pumped into the lymphatic system. Whenever there is less liquid tissue fluid, lymph fluid will come out and it will make up the deficit tissue fluid quantity. So likewise, lymph fluid and blood are separated in our case in closed circulatory system animals. But here, the tissue fluid is mixing with the blood coming from this blood vessel. So here, blood and lymph both are mixing. So that's why it is called as hemolymph. So in open circulatory system, there is no blood, there is no lymph. Both are mixed. So that's why it is called as hemolymph. So this is the difference. This is the structure and functioning of open circulatory system. So what is the disadvantage of this open circulatory system? Let us see in next slide. So again, let me explain one more about this closed circulatory system. So in closed circulatory system also, we have heart and we have arteries and we have veins. So our heart is collecting blood from the tissues through these veins. And again, heart is pumping blood through tissues through these arteries. And at the tissues, these arteries and veins both are connected with these small blood capillaries. So here, blood circulatory system is nowhere open. All these interconnected. So blood will be continuously flowing inside this system only. So if we carefully dissect the body of an closed circulatory system animal, we can never find blood. Why? Because blood is completely circulating in this blood vessels only. So, but usually we see that whenever we have a cut on our skin, we will find the blood oozing out from our that wound. So why it is happening? Because as the cut is made, the cut has disrupted these blood vessels. So only when these blood vessels are having a cut, then only blood comes out of the vessels. So actually, it is not practically possible to cut our body, to dissect our bodies without causing a damage to these capillaries or blood vessels. Wherever you cut, there are either blood capillaries will be there or a big arteries or veins will be there. So total our body is occupied by these blood capillaries or arteries or veins, so that we can never make a cut without damaging these blood vessels. So as the cut is damaging these vessels, that's why only blood will come out. So if by God grace or if by any miracle, if we cut our bodies without causing a single blood vessel damage, without causing damage to a single blood vessel, we can find there's no single drop of blood. That is the beauty of the closed circulatory system. So in this thing, what is happening in closed circulatory system, hearts have been developed a lot. They have grown big in size and they have highly muscularized. So heavy muscles are there in these bodies, so that heart collects blood a lot and heart pumps the blood with high pressure, so that blood speed will be high in these organisms. Whereas in open circulatory system animals, the heart is very weak, it is not highly muscularized, and it cannot pump the blood with force or with speed. So as all these organisms which are having open circulatory system, they are small animals or less developed animals like insects and some molluscans and some echinoderms. So all these animals, they do not require high pressure blood circulation. So that's why having the open circulatory system is serving their purpose. And coming to the advantages and disadvantages of these two circulatory system, advantages of open circulatory system. In this thing, exchange of material is direct between the hemolymph and the tissues, and there is no diffusion barrier. So here the exchange of materials takes place with ease. So easily the thing will be done. The exchange of materials will be done here in this case. But here in closed circulatory system, what is happening? The materials, food material have to, first they have to enter into the, say, let me explain with this diagram. Here, see, the blood has come to this tissue. So blood is flowing in these blood vessels. So the food materials, they have to enter into the cells of these blood vessel walls. And again from cells to the food materials have to enter into the tissue fluid. So here 2 times it has to cross some cells. So some barrier is there. So here in the open circulatory system, that barrier is not there. Blood is directly coming in contact with the hemolymph. So food materials easily they have access to tissues to get into. But here first it has to cross the blood vessel walls. After crossing the blood vessel walls, then only it enters into the tissue fluid. So somewhat it is some difficult when compared to this thing. And muscular walls of blood vessels, sometimes they can constrict and dilate to vary the amount of blood flow through specific vessels. This is also one advantage. So depending on the needs, when the tissue requires lot of blood, it will dilate and more blood will and more fluids will enter into the tissue fluid. When the tissues do not require lot of materials, then the blood vessels will constrict. So depending on the situations, high amount of blood circulation will be made and low amount of blood circulation also will be made. So that mechanism is present in these animals which are having closed circulatory system. Coming to the disadvantage, so control is not there in these organisms over the distribution of hemolymph. So to which parts have blood has to be sent, all these material, all these things cannot be controlled by the animals, and no mechanism for reducing the flow to a specific part of an organ. So as we have seen here, blood circulation can be increased or decreased depending on the needs, but that thing is not possible in these organisms. So this became a disadvantage in these organisms. And we have seen this advantage in this thing, material of exchange of materials is easy in these organisms, but in this thing, exchange of materials is somewhat complex. So lot of energy is required for this blood distribution and exchange of materials also. So, and another advantage, this thing, a fine scale control over the distribution of blood to different body regions is possible. So here in this thing, control is not there, but here in this closed circulatory systems, control is there. So these are the advantages and disadvantages of the open circulatory system. On the whole, closed circulatory system is highly developed over the open circulatory system. That's why highly developed animals have developed a closed circulatory system. And another topic is myogenic heart and neurogenic heart. And you must be familiar with this cardiac conduction system. You must have studied this in your second year intermediate course. So in cardiac conduction system, we have SCA node, AB node. Okay, and from SCA node, some conducting muscle fibers, special type of muscle fibers are distributed throughout the auricles. And these conducting fibers are connected to AV node, and from this AV node we know that this bundle of His and Purkinje fibers will come, and these Purkinje fibers are distributed throughout the walls of these ventricles. So in this way, here we have a conducting system. Starts with SCA node. SCA node is giving many conducting fibers. These conducting fibers are connected to AV node, and from this AV node again, bundle of his fibers and Purkinje fibers are coming. So all these things are distributed throughout the walls of the heart, throughout the artery wall, auricle walls, and throughout the ventricle walls. So here, this is the this type of heart is example for, it is called as myogenic heart. So in this thing, this SCA node is nothing but it is made up of some special muscle cells, and the heartbeat will be started by this SCA node only. So SCA node is starting the heartbeat. That's why it is called as pacemaker. As the pacemaker is a muscular structure, this type of heart is called as myogenic heart. Genic means producing or generating. So what is generating here? Heartbeat is generated here. Heartbeat is generated by a special type of muscle cell. So myo means it is related to muscle. Myo word is related to muscle. So SCA node is a special type of muscular cell. It is made up of special type of muscular cells, and these special type of muscular cells only, they are generating the heartbeat, and the generated heartbeat is conducted into this auricles through this green color fibers. They are conducting fibers. They are some special type of neuron-like things. Okay, so they will send these new signals in the form of action potentials. So all these things will be distributed throughout the walls of the auricle, so that auricle walls, they will contract. As a result of this contraction, systole of auricles will start. So when the systole of auricle starts, the heartbeat also will start. So heartbeat will start with the contraction in the auricles. So first phase in the heartbeat is contraction of auricles or auricular systole. So auricular systole will be triggered or stimulated by the SA node. SA node will generate electric signals or action potential like signals. Those signals will be sent to the muscle walls, okay, muscular cells of this auricle wall through this green color fibers, conducting fibers. So after receiving the signal from these green fibers, all the muscle cells which are present in the auricles, they will contract. So auricular contraction or auricular systole is being initiated by SA node. And once the auricle walls are completed their contraction, auricular systole completes. By that time, the signal will be received by this AV node. Now this AV node will supply this signal in the form of action potential through this bundle of His and Purkinje fibers to the muscle cells which are present in the ventricles. So upon receiving these signals from this bundle of His and Purkinje fibers, now ventricle walls also they will contract. So in this way, muscular contraction of this heart walls is being first initiated by SA node. So this is called as myogenic heart. Why? Because heartbeat is initiated by this SA node only. That's why SA node is called as pacemaker. This is the first pacemaker, and the second pacemaker is AV node. So here normally we know that heart must be functioning at every time. So and usually brain will control the heartbeat functioning, but here as the brain is busy with some other things, brain has given this duty of controlling heartbeat to SA node only. So whenever there is no specific requirement, whenever there is no specific need, SA node only will control the heart wall contractions or heartbeat. Okay, when you are in normal conditions, brain has no function in controlling the heartbeat. But in some special conditions, you require a lot of blood supply to the tissues. At that time, heartbeat must be increased. So during that conditions only, brain will take temporary control. So say for example, when a snake is coming to bite you, then what will happen? Your heartbeat will increase. Heartbeat rate will increase. Blood circulation will be increased. Blood pressure will be increased. Why it is increasing? Because as you have received the information, as your brain is received the information of a snake is coming to bite you, then your brain will realize that if it bits you, you may die. So as a result of that thing, brain what will it will do through the sympathetic nervous system, through the vagus nerve, it sends information signals to the SA node to increase the heartbeat rate, so that usually our heart beats 72 times per minute. Okay, but in this type of situations, our heartbeat rate increases beyond 72 times per minute. It may go up to 100 beats per minute like that. So when you require a lot of energy, when you require some special conditions like this, fight or flight hormones are also there. So when somebody comes to beat you, when you are indulged in a quarrel or fighting with your friend, then also your heart beat rate will be increased, blood pressure will be increased. Why? Because when you are fighting with someone, your muscles require a lot of nutrients, lot of oxygen. That's why only under such situations, your heart beat rate will be increased, your blood pressure will be increased. So at such times only, brain will take control. Under normal conditions, brain will do nothing in generating heartbeats. Under normal conditions, SA node only will control the heartbeat functioning. So that's why as the heartbeats are generated, controlled by this SA node, this type of hearts are called as myogenic hearts. And another type of hearts are neurogenic hearts. These neurogenic hearts are present in almost all arthropods and some molluscans. Except these organisms, in remaining animals, we have myogenic hearts only. So in neurogenic hearts, what is happening? Nervous system is controlling this heartbeat rate. Nervous system is controlling the heartbeat rate. So here in this thing, we can see this. This is a crustacean. It is called as squilla. It is one type of crab-like organism. So in these organisms, this pink organ is heart, and we are seeing this heart from the above view or dorsal view. So in this heart is supplied with a special type of nerve ganglion. These are called as cardiac ganglion. So these cardiac ganglion are they are directly connected to the brain. Nerve ring will be there around the esophagus in these organisms. So this nervous system is sending some special nerves to this heart, and this special nerves are forming a cardiac ganglion over the heart body. So these cardiac ganglion only, they will generate the signals to start the heartbeat. So as in these organisms, some special neurogenic organism, special neurons forming a cardiac ganglion, and this cardiac ganglion only sending signals to start the heartbeat rate. So that's why this type of animals heart is called as neurogenic heart. Now coming to the differences between this neurogenic heart and myogenic heart. In neurogenic heart, heartbeat is initiated by a ganglion situated near the heart. And in myogenic heart, heartbeat is initiated by a patch of modified heart muscle called as SA node. And in this neurogenic heart, impulse of contraction originates from the nervous system. Here, the impulse of contraction originates itself in the heart by this SA node muscle fibers. And in neurogenic heart, the heart normally stops beating immediately after the removal from the body. Therefore, heart transplantation is not possible in these organisms. Why? Because to start the heart beating, nervous system is required. But when the heart is separated from the body, it is separated from the nervous system. When there is no nervous system connection, heart stops functioning. That's why it is not possible. But here in myogenic cases, heartbeat is started by this pacemaker or SA node, so that even though if you remove heart from the body, heart will start functioning for some time. So within this time, if we transfer the heart to another patient, heart transplantation is possible. That's why in some cases, heart transplantation is made success because of myogenic heart only. And examples for these neurogenic hearts are some annelids and most arthropods. And this thing, myogenic heart, examples for myogenic hearts are hearts of molluscans and all vertebrates. So this is about the neurogenic and myogenic hearts. With this, we finish today's topic, and next we meet with another topic.